I'm Nate Upham, assistant professor in Souls here to introduce Bill Murphy, who's here coming. He's here from Texas A&M. So I'll try and be brief here. Bill is a professor of Veterinary Integrative Bioscience, which is a really cool title, actually. Yeah, and he's in the School of Veterinary Medicine and Biomedical Sciences at Texas A&M. Bill got his bachelor's at Illinois State, and then I was just learning, moved from Illinois State to University of Tulsa for his PhD, following his advisor who was starting his new lab there and was working on killifish genetics for his PhD. So he was not a trained mammologist from the start, but then kind of picked up the mammalogy reins. Yeah, there you go, forced marriage. And during his postdoc at the National Cancer Institutes in Frederick, Maryland. And then in 2004, he started as an assistant professor at Texas A&M. So it's been exactly 20 years since he started his lab at Texas A&M. And during that time, he's graduated 3 master's students, 5 PhD students, had 3 different postdocs, and 40 undergraduates have made it through his lab. And he has 8 current undergraduates as well. So there's a lot of training going on there. Bill is currently funded by the National Science Foundation Systematics and Biodiversity Cluster to do some of this work. He's had many previous grants. I looked and he has over 150 peer-reviewed publications, including, I think my favorite title was the one called A Dog's Breakfast, ? which was a perspective piece in Science, but that's a great title. 22 different papers Bill has in Science. That was the count I made, 3 in Nature, including 6 papers with over 900 citations. So no small feat, which has helped earn him AAAS membership in 2020. And then as of 2 days ago, on Tuesday, was elected to the National Academy of Sciences. So I think we need a round of applause for Bill.
Hey, I hold that applause till after I'm done. So we'll see how I do. So it's a real privilege to be here in front of some good friends and colleagues, talking about some of their favorite things, sex chromosomes and mammalian genomes. And of course, I hope I won't offend somebody if I say mammals are arguably the most diverse, phenotypically diverse group of vertebrates in size and form. Even in their genomes, I don't think there's a close competitor. They just have this incredible morphological diversity. They have wings, hooves, claws, scales. They are terrestrial. They're aquatic. They fly. They dig holes in the ground, and they have these amazing phenotypes like cancer resistance, echolocation. There's just so much phenotypic variation that it's exciting to be a biologist working on mammals to ask any number of different questions. And because of the fact that they have the same 19 to 20,000 protein coding genes, and we know this pretty confidently. Obviously, I'm hiding the fact that they have a lot of non-coding RNAs that make them a little different, and I'll dive into that somewhat today. But that inheriting that common set of genes yet has led to this amazing phenotypic diversity. And so having the ability to sequence and compare genomes really is just so powerful when it comes to trying to connect genotype to phenotype. And so my entrance into this field, as I was saying, was a long time ago, as a young postdoc at the National Cancer Institute, where I was told you could not work on fish, work on cats or mammals or humans. And so I was happy to work on some of the first cat genetic maps. And I was also lucky at the time, my postdoctoral advisor, Steve O'Brien, was invited to do the first genome issue on comparative genomics. So each year, this is obviously prior to the finishing of the human genome, they had a special genome issue about some topic. And they decided at the time it should be comparative genomics. And that was very much a cottage industry at the time. There weren't that many people working in comparative genomics. People worked on mouse and rat and cattle and dogs and cats and pigs, right? And so it was a small little group, but the NIH slowly realized that it was important to have those models to learn something about genome architecture even before we had genome sequences. So when I started comparative genomics, it was all about comparing chromosomes and comparing synteny before we even could study their genomes and study natural selection and changes in genome content and repetitive gene content. And so obviously after the human genome, which was finished a few years later, they asked, of course, how do we interpret the other 98% that's not protein coding, right? And so they quickly realized, well, we probably should sequence some other mammals like mouse, which is the premier mammalian model. But at the time, draft genomes were still incredibly expensive, $50 to $75 million per genome. So again, I feel really, unfortunate that I didn't have the cost for sequencing back then that we have now. Could have done a lot more interesting work, but how could we choose? What were the motivations? And so I was lucky at the time to be working on the phylogeny of placental mammals, and a lot of that was driven by this genome issue, because they tried to get all the paleontologists and morphologists at the time to agree on a phylogeny to put on the poster. And the morphologists quit. Seriously, they quit and wrote a nasty letter and said, we're not going to do this. We can talk about that later. And so, but basically the phylogeny we proposed that has pretty much stood the test of time, sort of revamped the way morphologists and mammalogists had thought about how organisms are related to each other. And so we learned surprisingly that things with hooves and that eat insects evolved multiple times and on very different continents. And so then the question that we posed and was starting to be asked at that time by NHGRI is, well, how do we use this information to better inform human biology and understand the human genome? And so we wrote this article in Science later that year called On Choosing Mammalian Genomes for Sequencing. And so that caught the attention of Eric Green at NHGRI, who came up the week after this paper was published to say, how do we pick? And so obviously biomedical relevance, economic importance were some of the first things that were chosen. That's why mouse and rat were done first. And then how phylogenetic close are you to humans? So pick the chimp so we can identify those changes that are unique to the human lineage. And then being phylogenetically distant from humans was valuable because that would help us understand what was constrained over time and could help us understand the evolution of regulatory elements. And so the motivation to annotate the human genome really drove probably, I don't know what percentage of that motivation drove this drop in the cost of sequence or the motivation to make sequencing more affordable. But as sequencing dropped, the number of genomes began to climb. I think we went through sort of a dark period here where, at least for mammalian genomes, we were able to sequence many genomes, but not to really good quality, right? And so there were a lot of fragments and there were a lot of misconceptions that came out of those earlier days. But now we're to the point where we can strive for perfection. And I'll talk a little bit about our work and our desire to actually have T to T or telomere to telomere or near gapless assemblies so that when we're trying to connect genotype to phenotype, you have all the information you need with regard to the genotype, right? Because if you have gaps and hundreds or thousands of gaps in your genomes, you don't know what you don't know. And I'll give you an example of that. And so I was thrilled to be involved in the Xenomia project. I know Eleanor came and spoke last year around the time this came out. And this was really the culmination of using comparative genomic sequencing to understand the human genome. I'm not going to talk about the Xenomia project today, but one of the most important outcomes is the ability to measure genomic constraint, these phylo-P scores, where you can, in any base in the human genome, measure whether it's neutrally evolving or under strong purifying selection or positive selection. And even more powerfully, because this is not a reference-based alignment, it's a hierarchical alignment, you can project those scores onto any species you're interested in and say, okay, in cats, what parts of their genomes are under constraint? Is it different from humans in some places? Where are they evolving really rapidly? So it's a really powerful tool. And I'm not going to talk too much about that today, but what we were able to do is really better understand and resolve some controversies with regard to the mammalian phylogeny. We can study individual gene histories. We're able to finally start parsing, instead of combining all the data, start looking at how the evolution of mammals differs across chromosomes and the role that natural selection might play. And ultimately, we made some conclusions about how and why the KPG extinction may or may have not influenced how mammals radiated. But I'm not going to talk about that today. But it was probably George Gaylord Simpson, perhaps the most influential paleontologist of the 20th century, and a major participant in the modern synthesis, who said this quote that is one of my favorites and really motivates me in my lab to push to more complete comparative genomic analysis, is that in 1945, this is the paleontologist who tossed out the genome paper in 2000 or 1999. The stream of heredity makes phylogeny in the sense it is phylogeny. Complete genetic analysis would provide the most priceless data for mapping of this stream, and he couldn't have been more prescient in that conclusion. And so I'm going to talk today about complete genomes, not necessarily T to T, but why having high quality chromosome level assemblies has allowed us to really dig at some longstanding questions that we certainly weren't able to do 2 decades ago. So I'm going to talk about why do complete genomes matter. I'm going to start and primarily talk about my work on cats and tell you why cats are a really great model system. We have these fantastic hybrids we can use to study speciation and how reticulation influences the genome and its patterns. And then we'll talk about how we've stepped back and used the phylogeny of mammals to study how recombination has evolved. And then I'll end on some interesting patterns that we've come across that we don't fully understand, and we'll be interested to get other people's opinions on this, is why mammalian chromosome, the X chromosome, is so important to speciation. So why cats? Well, there's a lot of people who are interested in cats for different reasons. Feline genomes are of interest to veterinarians and scientists seeking the genetic basis for disease. In the Cat Genome Project, I've been part of this 99 Lives Consortium. We've been able to identify many dozens of mutations that cause diseases that have analogs in human medicine. Breeders and geneticists, of course, are interested in the genetic basis of coloration. And so cats have this morphological pelage variation that you don't see in mice, and particularly in patterning and spotting. And also understanding the evolution of wildcat species. Initially, we took advantage of the domestic cat genome to study their genomes, primarily to understand the impacts of plummeting genetic diversity on fitness and conservation management practices. And so let me just introduce you to the cat family Felidae. There's about 42 species that are described, and this is a current phylogeny as we think we understand it. It is not completely bifurcating, but this is what we think is, if there is a bifurcating history, this is the closest to it. It has 8 lineages. Many of these are familiar to you. The big cats, Panthera, here at the bottom, the tigers, the lions, the leopards. And at the top, we have the domestic cat lineage. They all coalesce to about anywhere between 12 and 15 million years. And there's these 8 radiations. And for the longest time, we tried to build the phylogeny of cats to understand trait evolution. And we actually could consistently identify these 8 clades, but it was very difficult to resolve the relationships within the clades and even between the clades. The other thing I'll point out here is that our genome resources, because of the value of the domestic cat as a biomedical model, is you see this column here. These numbers are the number of raw whole genome shotgun sequences for any of those species. So you can see in the domestic cat, we have 583 currently domestic cat genomes that have been sequenced, and all the way down to even 287 tigers. And so we have a lot of population genomic resources, but we also have excellent reference genomes for many of these species. And shown here is the N50, both contig and scaffold. And you can see that for many of these, our contig N50 is almost the same as our scaffold N50, which means we are near gapless, and that's become extremely powerful. And I'll give you some illustrations of that today. But as we started to parse genomes and not sort of build concatenated phylogenies of cats, but we started to look at how the history is varied, we learned that cats' genomes are a mosaic of gene history. So what I've shown here is an exemplar of each of the 8 lineages of cats here on this axis, y-axis, and then that's basically each row is the genome of each of those cat lineages divided up into 50 kilobase windows. And so this is showing us where along the chromosome, the species history within the Panthera lineage, where we think the species tree is located, and those would be in white. And anywhere we see a strongly conflicting phylogeny from what we infer to be the species history, that's shown in gray. And so you can see there's a lot of phylogenetic discordance across the genome in different lineages, more in some than others. And there's ecological reasons for some of those differences. And even more controversial is the fact that we now see that every distinct mode of inheritance, so the mitochondria, the X chromosome, the autosomes, the Y chromosome, all produce a different phylogeny between the different clades. And why is that? And unfortunately, most phylogenetic methods that we've used for a long time assume some underlying majority rule consensus criterion, right? And so they assume the true signal, the true phylogenetic signal should be the predominant signal, that we see in the genome. And we published a couple of years ago our first whole genome-based approach of the cat phylogeny. And what we showed is a lot of that discordance I just showed to you was not due to random genetic processes like incomplete lineage sorting. Most of it could be explained using statistical tests that infer introgression in the past. And this is held up as we've increased the quality of the genomes and increased the quality of the alignments. But basically, cats have had this immense history of ancient and ongoing hybridization that we see today. For example, along the US-Canada border, that's where the bobcat meets the Canada lynx. And they have a stable hybrid zone that's probably evolved over years. And we know that they produce interspecific hybrids. And so there are many other examples. My colleague, Eduardo Isaric in Brazil, studies a hybrid zone there in the Leopardus genus. So initially, this idea that hybridization played a factor in speciation or even genome architecture was quite controversial. But now we know that it's not rare at all. It's all over the place. And in more and more mammal groups that are looked at, and even across the tree of life, butterflies and plants, we see that hybridization is not rare, and it might even be the norm in many groups. And so for phylogenomics, this is actually a challenge to some of the most widely used phylogenetic algorithms. So if we look at 3 examples that came out in the last 10 years, some really good window-based phylogenetic approaches to looking at how discordance happens across the genome with mosquitoes, cats, and butterflies, we see that across the genome, this phylogenetic signal is really spatially variable, and each of those colors shows a different tree topology. You can only see this by analyzing tens of thousands of windows. But if we decompose these data a little bit into yellow, what we infer is the species tree, or perhaps an earlier set of branching events, a bifurcating component, relative to trees that either reflect ILS or ancient hybridization. And if you place those in this context, you see very clear patterns. One of those that I'll talk about today is the fact that the sex chromosomes, the X and the Z chromosome, is enriched for the species tree. And you also see this pattern, and Edelman was the first to show this, and we saw this in cats, and many other lineages of plants and animals show the same pattern, is that the distribution within the genome is predicted by a recombination rate. And the non-species tree in green is more frequent in high recombining regions, and the species tree is most frequent in low recombining regions. And we see almost the exact same pattern in cats. We see this, again, in many, many different lineages. And this sort of makes sense. We know in low recombining regions, there's going to be reduced effective population size, so less ILS. And then high recombining regions are more permissive to the retention of introgressed foreign alleles, if we want to call that, from introgression, because they're more readily unlinked from negative epistatic or potentially negatively epistatic interactions. And so the discordance doesn't come as a huge surprise. Like I said, we knew from natural field observations that cats hybridize. And so this actually depicts sort of the propensity for cats. I'm not even showing all the examples. These are the ones that I actually believe there's good literature that within either captivity or in the wild, that cats can hybridize actually across extremely large phylogenetic distances, evolutionary distances. And so turns out cat hybrids have been extremely powerful for cat genomics and the development of cat as a biomedical model. So the Bengal cat was actually used for the first genetic maps. When I was at NCI, we actually had a colony of cats, excuse me, and so the other nice thing about having hybrids is they follow these rules of speciation. So cats follow Haldane's rule, which states that when in the F1 offspring of 2 different animal races, One sex is absent, rare, or sterile. That is the heterozygous sex or the heterogametic sex. And so they follow Haldane's rule. Interspecies hybrids, the F1 hybrids, the females are fertile, the males are sterile. These breeds that I just showed you are performed by back crossing those F1 hybrid females to domestic cats. And eventually those cats, those hybrid cats, regain fertility, and then they establish the breed through intercrossing. And that becomes important in a minute. They also exhibit the large X effect. And the large X effect is this common observation, not just in mammals, but across, again, Drosophila especially, and especially a lot of studies by Jeff Good and his group in rodents, that there's disproportionate enrichment of large effect genes that drive reproductive isolation. And so they accumulate on the X and even on the Z chromosome. And so cats have these hallmark sterility phenotypes that we also see in rodents. Most notable, they can show, like F1 hybrids show more severe meiotic or pre-meiotic arrest. But the most classic is shown here is this meiotic or pachytene arrest that's actually seen in rodent hybrids, cattle hybrids, most mammalian hybrids. That's what you classically see. And then post-meiotic arrest happens as well. So one of the biomarkers that was first observed in rodent hybrids was that there was this upregulation of the X chromosome seen in sterile hybrid males in their testes. And the reason for that is in meiosis and at that pachytene stage, the sex chromosomes become condensed and form this sex body in which they are silenced. And so something about incompatibilities on the X chromosome disrupts that silencing and leads to inappropriate expression of the sex chromosomes, which leads to this massive apoptotic response and sterility. And what we see across cats when we've studied gene expression in sterile versus fertile hybrid testes, you can see consistently the X chromosome is upregulated with respect to the autosomes. The Chowsy, interestingly, has a massive downregulation of the autosomes that's interesting. We don't fully understand that, but that's for another talk. And so to really understand what is the genetic basis for this large X effect, we started using some of our hybrid crosses. And so we used one, the most closely related to the domestic cat, to the jungle cat. They diverged about 3 million years ago. They were bred to create the Chowsy breed, which after 4 to 5 generations of back crossing, these males and females are interfertile, so they can interbreed those and create this cat that looks kind of exotic. That's the goal of most of these exotic breeds, as they're called, is to create a wild-looking cat that someone will pay $10,000 to $20,000 to have in their home. Right? For the record, I don't own one, and I don't want one. So, but I'm happy to take the samples from breeders. And so here's our first GWAS, where we genotype about 30 sterile and fertile Chowsy hybrids that are almost all from the 4th to 6th generations, and they all have about the same amount of wildcat ancestry in their genomes. And we had this very strong hit on the X chromosome. We were able to fine map that actually using, they're not showing up, but we had ancestry informative markers spanning the X chromosome due to sign insertions that were specific. And so we could call, for example, in sterile hybrids, they all had this interval here that was common across them on this region, around 90 to 100 million base pairs on the X chromosome. And we're able to fine map that down to a 500 kilobase critical interval. And we zoomed in the genome assembly we had at the time. This was almost 8 years ago. Big old assembly gap. That really sank. And it sat there through multiple upgrades and iterations of the cat genome. So we were kind of stuck. But we pushed on because there was some really interesting biology that was coming out in the literature at the time. So there's our top GWAS, and there's the gap. On the edge of that gap was a tandem repeat. And that explains why the assembly broke. Assemblies tend to break anywhere there's a lot of polymorphism between the haplotypes, right, or heterozygosity. And macrosatellites, not micro, but macrosatellites where the units are several kilobases in length, they are prone to break, much like many other things like centromeric satellites, right? We never were able to sequence those until very recently. But the candidates, PLS3 and the one on the other side, weren't good candidates because one of them was not even expressed in the testes, and the other one had no known role in meiosis or had ever had a fertility phenotype in mice or others. but we were very interested in DXC4, and we knew that's what it was, DXC4. It's not a protein coding gene. It's a tandem repeat that has non-coding RNAs, and it had been studied in humans, and I'll get to that in a minute. And so what is unique about macrosatellites? Their biology is quite interesting, and they've been largely missing from a lot of genomes, even they are known to cause a number of diseases. They have large multi-kilobase repeat units. They're highly variable in copy number. They're enriched for CPG islands. And they have this interesting copy number threshold mechanism for methylation, whereas above a certain level, they may be hypermethylated. Below that, they may be above a threshold, they might be hypermethylated. Below that level, they may be hypomethylated. And we know in human studies that changes in those thresholds that's very distinct to different satellites can cause silencing of the regional genes in the vicinity of those satellites. DXC4 was interesting further because it was implicated in female X chromosome inactivation, which is a silencing mechanism in which one of the two female X chromosomes is randomly inactivated. And so what you see is in a normal female active X chromosome, this is a high C map where these triangles are showing interaction matrices. And this is typical of what you'd see on an autosome, and the active X chromosome, where most of it is euchromatin and is actively expressing genes. There are parts that do stay silenced, including DXC4, so it's inactive on the active X chromosome, but it has the opposite conformation on the inactive X chromosome, where it and a few other partners escape inactivation. They're all repetitive, they all encode non-coding RNAs, and they superloop in three-dimensional space and interact and help, it's thought, to localize the bar body, the condensed inactive X, to the nucleolar membrane. And so we'll come back to those loci. Fire, DXC4 exists, and another one called ICE. They're spread throughout the X chromosome, and like I said, they superloop, they interact in 3D space, and that might become quite important when we start thinking of the evolution of the X chromosome, some of the patterns I'll show you later. And so DXC4 forms this hinge group, where when you look at high C data from an inactive X chromosome, it forms these superdomains, right, that are largely silent structures. Now, why are you so interested in DXC4 if that's a female phenotype? You're talking about male sterility. Well, turns out in males, as I said, the hallmark of male sterility, this meiotic arrest, occurs at a time when the XY body is supposed to form. And that's another mechanism where the sex chromosomes are silenced. And we don't really know fully how that is formed, but we speculated that perhaps DXC4 might play some role in that process. And one of the reasons for that is there were mouse studies showing that the same regions on the X chromosome escape X inactivation or escape silencing in both male spermatogenesis and female somatic cells. And so that made us think perhaps DXC4 is doing something. So we're still stuck. We have a gap in our assembly. What can we say about DXC4? So along came a new procedure called trio binning, developed by Sergei Korin. And this really came along at the time of long reads to basically be able to phase the assemblies or phase the haplotypes of a single diploid individual. So this works really well in hybrids, especially the more divergent their parental haplotypes are, the easier it is to phase the reads. So we generate long sequence reads, in this case an F1 Bengal cat, and then we sequence with short reads the parents. And then those parents, we identify unique K-mers that can then sort the original parental haplotypes, their long reads, into independent pools before we even try and assemble them, so that no longer there's this confounding issue of mixing haplotypes and breaking chromosomes as a result of the assembly process. So what we were able to show when we published this last year in Nature Genetics is this trio binning was extremely effective. We did this across 3 different F1 hybrids, the Bengal cat, the Safari cat, and the Liger. And these data I'm going to show you are based on older, noisier CLR or circular consensus sequence. Doesn't have the accuracy of Hi-Fi. I'm not going to show data today, but we've done and started following up on all of these using Hi-Fi technology and ultralongs to get us truly or nearly to T genomes in cats. And as you can see at the time, this is a fairly old slide, but you can see our assemblies were as good as any other domestic animal genome, right? In terms of the number of gaps, less than 100, close to 50, and contig N50s that were extremely high. Of course, what were we missing? Well, if any of you read the human T to T papers, you know, centromeres and telomeres are now there for us to look at and study their evolution, and they evolve really rapidly. Complex gene families like MHC class 1, immunoglobulin heavy chains, olfactory receptors, which I'm not going to talk about today, but we had some cool findings between cat species about how those regions of the genome evolved. And then large tandem arrays like DXC4. So back to the DXC4 story. We have cat finally resolved the structure, and sure enough, it's its long tandem repeat. Here's the human DXC4 that was only first finished in the T to T assembly. And it's a little different. You can see actually cats have two arrays, an A and a B array, where in humans, there's one CTCF binding motif within each unit, in each array unit. And cats have one that's like that, but then they have another one that actually has opposing CTCF binding sites. We don't know what those two repeat arrays are doing, but they actually evolve quite differently. But most relevant to our phenotype was my graduate student, Kevin Bredemeyer, who was tackling this hybrid sterility project. He was able to show for the first time that DXC4 was actually expressed at the time the X chromosome should have been silenced. So it suggests DXC4 did escape silencing, and it was expressed in whole testes and pachytene spermatocytes, the stage that the X should be silent, and in round spermatids. You can see we have these transcripts. We recovered these two transcripts that are extremely divergent from, but are orthologous to human DANT1 and DANT2, the transcripts that DXC4 produces and helps superloop with those partners. And we compared the methylation frequency of fertile and sterile hybrids. And our expectation was, is indeed, if there's a copy number mechanism, if there's a threshold, then perhaps if there's a different size in a jungle cat haplotype within an otherwise domestic cat background, it might be causing problems. And sure enough, we show that in the methylation frequency in sterile chowsies is significantly hypermethylated relative to fertile chowsies and fertile domestic cats. So this was only in the first repeat unit A. We didn't see an effect in repeat unit B. So this made us think repeat unit A has something to do with this phenotype. And it also matches the observed downregulation of DXC4 that we saw in the sterile hybrids. And so it's interesting just to show that, again, it made sense, our model, because the jungle cat had more copies than the domestic cat. So we speculate, we don't have strong evidence for this, that that lengthening perhaps is leading to hypermethylation in the background, possibly because of interactions between the jungle cat haplotype on DXC4 and maybe domestic cat alleles elsewhere on the chromosome. That's something we're currently exploring. But even more important, DXC4 is evolving super rapidly, much faster than the rest of the genome. And many of these repeats that we're now able to sequence, like centromeric repeats, evolve extremely rapidly. And what you can see here, so these are the two repeat arrays. These are stained glass plots that basically measure divergence between highly repetitive satellite arrays. And this is all comparison to the domestic cat. And you can see a domestic cat, they have about 98 to 99% sequence divergence across the arrays. But as you move out in the phylogeny and make comparisons across them, you can see the arrays diverge extremely rapidly relative to these non-coding intervening sequences. And in fact, when we measure and compare how fast is DXC4 evolving relative to the rest of the genome, so if we divide up the genome, calculate genome-wide p distances between the domestic cat and Joffrey's cat to tiger, this is the rest of the genome. You can see DXC4, RA, and RB are way out there. They're evolving much faster than the rest of the genome, and that makes sense, that they would be a template, a rapidly evolving template upon which hybrid incompatibilities may evolve. So that was our first clue into a potentially interesting mechanism causing hypersterility in that cat breed. But we also found in the X chromosome that it actually was an outlier in terms of its structural variation. Now, I didn't show you this slide, but cat chromosomes are really boring. The karyotypes are almost all identical. But if we start looking at structural variation, the X is an outlier. And in fact, the inversion sizes are consistently longer on the X chromosome than are found on the autosomes. Most of these inversions are less than 2 million base pairs. They're all very small. And this is showing you different branches on that phylogeny of 5 cats that I showed you. X is always larger. More interestingly is where the inversions are found. They are found in a large recombination cold spot in the center. This is almost 50 million base pairs in the cat. This is actually the tiger. The cat, domestic cat recombination map looks literally identical. There's no difference whatsoever. And you see an enrichment, specifically fixed inversions, they have twice as high as protein coding gene content as those that are polymorphic. So that's expected. And in fact, there's a study in primates that came out around the same time that showed that inversions in humans and apes were also enriched and toggling within the same region of the X chromosome. And then back to our phylogenetic signal. Why is the X chromosome strongly enriched for the species tree? Well, it turns out if we start looking at the phylogenetic histories in cats, where we have these three top most frequent trees in the phylogeny, blue is tree 1, the most frequent genome-wide, tree 3 or tree 2 or tree 2, you can see that the most enriched tree is not the most common in the genome. And in fact, we infer this to be the species tree. And we have statistical evidence that these blue topologies are actually introgression trees. And those topologies are much younger than what we infer to be the species trees. But you can see the patterns almost identical across 3 different lineages of cats. So that sort of follows the large X effect. Something's happening in there and some of these other cold spots that is repelling introgression and perhaps holds the promise of genes involved in speciation. Well, Gang Lee, my postdoc who worked on this at the time, those phylogenomic studies, showed that actually cats are not the only ones like that. Pigs and humans also show a very similar X chromosome landscape. I'll remind you that their X chromosomes are collinear. The gene order in human, pig, and cat are the same. There are no inversions that can explain these recombination cold spots. They just exist. And so that led us to really start to want to understand the role of recombination and how it's evolved over tens of millions of years in mammals. And so we currently have a project where we're studying and using high-quality long-read genomes based on hi-fi and public databases to generate recombination maps across the placental phylogeny to understand, are these patterns conserved? Are there parts of the genome that remain historically low? Maybe those are useful for inferring phylogeny. What about those that have been historically high? Do they contain a lot of genes involved in adaptive evolution? So a team of undergrads with Dr. Nicole Foley started generating 20 high-quality chromosome-level recombination maps from across the tree of life. Show you a smattering of those here. They show the predicted relationship of larger chromosomes tend to have lower recombination rates and smaller chromosomes have higher recombination rates. And the X, as you'd expect, is an outlier, shown by those black triangles. What also is special about the mammalian X is it's highly collinear. The autosomes are scrambled. They're all over the place. But the X chromosome in most lineages is highly or has strong linkage conservation. These few exceptions, we actually have evidence that this bear assembly and this Rosetta bat assembly are wrong. And so they probably are also collinear. Any of you dealt with genomes and databases that aren't well curated have probably run into things like that. Anyway, but more strikingly is that recombination pattern is remarkably conserved across deep time. This is 100 million years of evolution. Now we're digging into this. As we developed these linkage maps, we started to look to see, are these patterns we saw earlier with the introgression tree being more biased towards the ends of chromosomes in regions of higher recombination. Sure enough, this is a group of bats and a group of carnivores, mustelids. And you can see if you look at the introgression tree, notice that the X chromosome is almost completely depleted of the species tree and the introgression tree is the X chromosome is enriched for the species tree and depleted of the introgression tree. And you'll also notice is that on the large chromosomes in the bats, near the centromeres, where there's low recombination, you have little introgression, and it increases as you go to the end, and it gets more frequent on smaller chromosomes. So we're seeing these same patterns over and over across mammals. When I first saw this, and Nicole showed me this slide, I was sure it was a mistake. I said, are you sure these are not two cats? This is the cat picture of the most frequent topology and what we think is the species tree. We can argue that over beers, but they almost show the exact same pattern. So there's enrichment in these same cold spots and retention of the species tree towards the middle of chromosomes. Why would that be? We actually look across more mammals, and we're starting to see that when they're collinear, the X chromosomes are the same, their recombination profiles shown in these blue tracks are remarkably similar, where the boundaries of this cold spot are almost identical across 90 to 100 million years of evolution, and the frequency of the species tree is extremely high, and in some cases, nearly fixed. What might be driving these patterns? I'll share with you a few ideas before I end. One that another graduate student, Wesley Bashir, showed by some of our first cat genetic maps is that, remember those interacting loci that form 3D structures? They are in the same position as you might expect in cats and pigs and humans, but they all have the same conserved linkage. Their gene order is the same. When you start looking at rat and mouse that differ from human, pig, and cat by 10 inversions that have happened, Pavel Petzer and his group have shown through those reconstructions that it takes at least 10 inversions to convert a mouse X chromosome into cat, human, or pig. But yet the spacing and distribution of those interacting loci are almost the same. And so we think that these inversions may have maintained this super looping potential so that important processes during X chromosome activation are retained. But also there's more functional landmarks. So blue here is our recombination cold spots, red are conserved hot spots. Right in the middle of this big blue one is the X chromosome activation center. And we know that interacts in 3D all the way throughout here. In these hot spots, I don't know why the others didn't show up. Of course, we have our pseudoautosomal region that's really high. It's always high. DXC4 is on the edge here of a cold and hot spot. Ice is over here. Turns out, for those who like sex chromosome evolution, this is about where the X conserved region is that's only shared with marsupials. It's about that same boundary. Also turns out, so those are potentially impacting female fitness. Turns out these same regions are enriched in male ampliconic testis-specific genes, that same desert in pigs and cats and probably under mammals. And so that makes us think, is it possible that some of the same genes or gene families are targets of selection, and maybe they're recurrently used in reproductive isolation within this landscape? So I'll leave and summarize, or leave you with some of these thoughts that I hope I've showed that haplotype resolved comparative genomics, they can reveal complex repetitive regions. They're not resolved in diploid-based assemblies. These are getting a lot easier and cheaper to do. This DXC4 macrosatellite, we think, plays an important role in feline hybrid sterility. We're now trying to test that in other hybrid crosses. So I have students looking at the looking at the Bengal and the Savannah crosses to see, are we also seeing DXC4 involved in that, or are there other loci on the X chromosome that are evolving very rapidly? And then we've identified this really large X-linked region enriched for structural variation that resists introgression across placental mammal history. And this is almost unheard of. I've not seen anything in another taxonomic group that resembles this level of conservation and conservation of the recombination patterns and the phylogenomic patterns. So we think analysis of this region may be key to resolving some of the more challenging questions in mammalian phylogeny moving forward. And so with that, I'd like to thank all of the folks in my lab and students who have contributed to the things I've shown here, folks at the University of Washington, especially Evan Eichler's lab for producing ultra-long reads that's helped getting us to T, my long-term colleagues in cat genomics at the University of Missouri, Leslie Lyons and Wes Warren, who've been working together on improving the cat genome, studying cat phenotypes, and then Mark Batcher and Jessica Storr, who are also involved in the repeat annotations of our cat genomes. And then, of course, NSF and the Morris Animal Foundation for generous funding. And with that, I'd be happy to take any questions.
Wow, awesome talk, Bill. Do you have any questions coming up? Yeah, John.
So that region you talked about at the end, with the sort of the three non-coding RNA regions that seem to be structurally near each other in various parts of the biology that I can't remember. I mean, I guess I'm wondering how much do you think in the lack of recombination? Like, I guess I'm wondering how much do you think of that is based on some? It sounds like most of you think it's structural in the sense that like, they have to be in the same order. How much of it, the lack of recombination, is also just rapid evolution of this region, and it's sort of, I guess I'm asking, is there an inability for recombination just mechanistically at the DNA level to happen after you get past a certain amount of divergence, right? So how much of it is driven by structure and how much of it is driven by seed?
Great question. So interestingly, you might suspect that, initially we thought, well, this has got to be some really large inversions or structural variations. So in a couple mammalian lineages now, we know that this is recurrently inversions that's driving this suppression. We have some evidence that's happening here. The macrosatellites, though, that we think are interacting, they're actually kind of separated by some of these boundaries, right? And so I think they're sort of, at least DXC4 might be buffered by fire, which is over here, and ice is right around here. And so we think that, to the reasons I said, I think that maintaining that order, because remember when you have these interacting loci, they have CTCF binding sites and they have to maintain a certain orientation. If you start messing with that, they can't form superloops. So I think the order and spacing is driven by that requirement. What that spacing, what that selection on spacing is, why does it have to be 40 million base pairs apart from XIC and DXC4 versus 20, and it doesn't work? We don't know, but we do see those consistent patterns. I'm not sure I answered your question really well, but
Maybe it wasn't a good question. We should move on.
okay. Yeah, so there's this sort of dichotomy between slow and fast, where you do have these really hot spots, and DXC4 is kind of on the edge of one of those. But you're right, it's the spacing. Those cold spots could be just there to maintain that spacing. But they also could be there to maintain these meiotic silencing processes as well. Yes.
Let's
go
to Melissa in the back first, and then
Weird, because X chromosome should recombine in females where there is no diversity. So, question: have you compared this structure and the recombination map between the therians and the marsupials?
Not yet,
Ooh, okay.
because we don't have good maps, so that would be intriguing. It does. We do know that the linkage conservation stops at placental mammals. So this part in they share all of this.
Yeah.
Their order is rearranged. They do have X chromosome inactivation, but it's a different mechanism. So far, we've not been able to find these types of repeat structures in their genome,
okay.
but their genomes are not quite T to T yet. So hopefully some new T to T genomes will help us answer that question. We did try to use high C data from some Monodelphis domesticus from one of my collaborators, A&M Paul Samelo. We were not able to see any of these structures, so we do think it's probably a placental mammal specific effect. That's my best guess at this point.
Cool.
Yeah.
all right. Yeah. Noah, do you want to already shout it out, or?
I have a lot of questions. I'm going to ask one simple one first, and then maybe a longer one. So the DXE4 in the inactive X sits right at like these large TAD boundaries, right?
Yeah, these superdomains.
These superdomains, right?
Yeah,
right.
This one's not.
And that's just in the just in the inactivated X.
That's right.
Is that across all tissues? Do we know? Like, I mean, I know
I
an
think
X,
so,
X
yeah.
is always one's always inactivated, right? Isn't it?
So
Okay.
But there's no. I'm pretty sure it is. We've only tested. This is fibroblasts.
Got it.
Most of the studies that I've seen in rodents are fibroblasts because they can actually use some weird mouse hybrid lines and make sure they're fully phased, so they can easily see this pattern. But presumably, yes, it's seen in other tissues. Yeah.
Cool. And just so I made sure I followed completely, the DXE4 macrosatellite is rapidly evolves. Why, we don't know. Like, it's just, it's a satellite, so there can be lots of shifts and changes, and after reproductive isolation, right, of populations, it rapidly evolves, and then because it's so important for these loops, that's what leads to this, theoretically, the sterility in
It could either be copy number variation driving that methylation,
or the order of the,
or it could just be raw sequence divergence.
right. So
We don't know. We don't know the answer, so I'm leaving that one open. That's
drifty evolution
speculating.
relative to the rest of the.
That's right.
Is it are there transposons that overlap with that? Like, do we know the origin of this DXE4 sequence?
I don't think so.
Yeah.
I don't think it's a transposable element. Another thing I didn't really point out is what's amazing is if you look at, I'll try to slide that. So DXE4 is conserved across mammals. It's for Melissa, but you can see it actually evolves pretty rapidly. So cat has both an A type and a B type. A lot of these genomes at the time were not complete, so we had to speculate, but you can see the A type mostly found here, or the B type in very few species we think share both types. But the repeat length, despite evolving so rapidly, maintains about a 3 to 4 kb length. Let's try that. I have no idea. Right? So that's something we don't talk about comparative genomics is spacing, conservation of spacing. Right? Physically on the chromosome, like I talked about, but why are these repeat unit lengths only being about 3 to 4 kb? Why aren't they 8 kb or 1 kb? Well, we don't see any variation or a lot of variation, but I'll admit these are not the best assemblies. I think with the newer hi-fi T to T assemblies, maybe this story will change a bit, but this is particularly interesting. Turns out we think it's absent in those species, but that's absent because I'm not convinced those assemblies are complete yet, but they're close. They're getting there. The ruminant T to T project is well underway. So that, yeah, cool observation. I don't have a great answer for that, but that's something, another layer of comparative genomics to think about is spacing and size. Why does that, what's the constraint?
I have a question. So it seems like it kind of changes, you kind of blow the paradigm wide open in a way of like, if you're trying to reconstruct a phylogeny of a given group, get genome-wide SNPs, do something like RADseq to, restrict to cut down the representation of the genome, and then it's just the dominant signal. Are you arguing that we should just be sequencing this one region of the X chromosome, and then that's going to like solve phylogeny?
The answer is yes.
Wait, so you're not arguing yet?
I think,
or you are, yeah.
I will go on a limb and say yes. That's one of the reasons why we're doing this recombination project in the first place, is we're trying to identify, are there places of the genome? So if you're using natural history collections, you can't sequence a whole genome.
Right.
Where can you target that's going to be the most likely to retain the species history, if there is such a thing, right? So turns out, I didn't show this here, Nicole has some data that shows that you actually use UCEs, which are a very common marker, they infer the introgression tree. The way they're distributed in the genome, they will infer the wrong tree all the time. So we actually think there's a lot of phylogenies out there that are wrong.
Well, for sure.
If there is a lot of introgression. Now, we have many data points where we've looked at lineages with these maps where the predominant tree is the same one on the X and the autosomes, but it always doubles down on the X. The X is always higher in frequency than the autosomes. So if you're going to have discordance, it'll be on the autosomes. The X, for whatever reason, tends to be less discordant. So I do think, yes, the X ultimately, some parts of that might be informative for not just phylogenetics, but also our future interest, something we're working on now, is trying to be able to find map across many lineages. Are the same parts of the X chromosome resisting genetic exchange? These are kind of coarse, these big blobs I'm showing you. Mm-hmm. If you actually look closer, I didn't show you some of our finer scale results based on really almost T to T genomes. It starts to pixelate, but we actually see cases where same regions in bears and in cats, they're much smaller than 50 million base pairs, but they might only be a couple megabase pairs, are really retaining that signal. And we think that it's probably these ampliconic genes, these Tesla-specific genes that are found in mammals. This is where we see a lot of the enrichment. We see some other enrichment sometimes over here. And we think that there might be recurrent use of these genes, maybe under positive selection, that are Tesla-specific and are found in all different mammals.
Really, really interesting.
Those would be good targets for phylogenetics, I think.
Yeah, Jay?
To what extent do you think that the introgress regions of the autosomes are being impacted by selection? Opposed to just
Great question. So there's actually a lot of good evidence for that. So we know if you look at human genomes and you look at where Neanderthal progression persists, there's good evidence that some of those spots that are enriched are for immunity loci. And I believe there's been, I don't remember exactly the papers, but they've done some functional tests to show that the Neanderthal allele elicits a stronger immune response. So we have seen some cases where adaptive introgression in cats in the big cat lineage is found in both. Well, in some cases it's in cold spots, but it's also in some hybrid combining regions too. So yeah, for sure, introgression can be adaptive. There's a lot of literature on that.
But I was sort of asking about the proportion of the introgenic strands that are impacted by selection. The ground limb, sort of what you've shown suggests that there are large proportions of these genomes where the gene tree is distorted species tree.
And how much of it's
due to introgression.
Yeah.
That is plausibly being impacted by selection in a strong way, would then suggest that a large proportion of the genome, at least the level of divergence, is actually being governed by selection, at least, link selection.
Yes, for sure, possibly. I don't think I could go out on a limb and give you a proportion, but maybe.
Percent.
50 seems like a lot. I would say maybe 5 to 10. I don't really have any data to hang on to there.
What proportion of the gene trees in the autosomes are distorted with the species tree? From your figures, it looked like it was, a chart.
Well, I will admit some of that is the way in which these, so you can see this looks like one big block, but you can see there's something there. So in this particular case, it can be as much as 70, 80% that are discordant.
Yeah, so
yeah, for sure. But that's not all. I'm showing you some extreme example. There's a lot of times where the species tree is the most common and introgression is still showing the same pattern. It's more limited, right? But there are certainly cases where there's almost complete replacement. The Anopheles mosquito example, I think, is a great one where they replaced almost all of the autosomes due to a very recent introgression between two malarial vectors. This was published by Fontaine et al. In Science in 2015. And that paper really got us starting to think about some of the patterns we saw. So again, that's wiped away almost 90% of the signal due to introgression. And that's like a no-brainer. That's not being able to infer introgression versus ILS or something else. It's clearly a very recent event. So obviously the fitness consequences of how much introgression isn't so great.
Great talk, Bill. Thank you so much. It makes sense that there's this region on the X in placental mammals. I'm interested in groups that don't have an ancient sex chromosome system. Perhaps you know of one. And so the autosomes are more interesting to me. Are there regions on the autosomes that you're seeing that are consistently giving you the species tree?
Yes.
And could you expand a little bit more on that? Are they just the centromeres, or is there something a little bit more universal?
No, I'm not going to say anything. Yes, we find spots. Is there an enrichment that's a smoking gun if we do gene ontology? No, but we're finding them. And at least in mammals, we're finding them. So then the question is, what do they look like in reptiles or birds? Are they the same things or not? I'm not going to, hold on. That's in the works.
Okay, I'm having some FOMO seeing all these great assemblies.
Where things are maybe not great for species, where we're seeing a lot of adaptive evolution, we do see some signatures associated with immunity, as I was just mentioning, not just in human, but in bats. This paper we studied, a paper we published in Cell Genomics earlier this year showed that introgression was enriched in immunity loci in Myotis bats, and particularly on microchromosomes because they have really high rates of recombination. So again, is that a general pattern? So I do think our hope is that this project, we have found conserved cold spots. There's some of them. And then what we really want to understand is how did those change with respect to characteristic evolution? Yeah, good question. We can talk more about that later. Yeah.
Any other questions? Yeah, Alexander.
Thank you very much. Thank you very much, Bill. So the cat and pig, as you said, are great examples because they show this really clear region of lower combination. But I believe you had a figure in which you showed other mammals, and it didn't seem as clear. Can you go over your rationale for saying that it's present in other mammals' placentals as well?
Right. So this is the way this is color coded. Is red is sort of hot relative to the genome-wide average recombination rate within that species. So in armadillos, you can see in elephants, we have the PAR is almost always very high, as you'd expect. And you can see there is a central cold region, so it's either devoid of any color. So it's basically this is cold, really cold, really cold. And again, these are collinear chromosomes. The gene order is the same; they're not rearranged. So you can see, and they're scaled so that they're almost the same landmarks are lining up. It's not perfect, but you can see, yeah, there's some exceptions where, what's going on in those old world monkeys? I don't know, but they're breaking the rule. They're not, right? So that's interesting, right? But yeah, so almost everything we've looked at looks like this. There are some other exceptions where, particularly when there's a lot of ILS in the genome, we don't really quite see that pattern. But when you have lineages that there's clearly evidence of hybridization happening, and that's the predominant signal, that's when we start to see the same sort of pattern. But it is, from the genomes we've sampled, it's pretty conserved. What we're looking at next is if you've got rearranged genomes. Do we see that same pattern? I don't think I have the slide with that, but it's not perfectly conserved, but it is broadly conserved. And again, these lineages go back to at least 95 million years in terms of their ancestry. So we don't think it could be conserved, or it could be recurrent. It could be evolving convergently. That seems unlikely. But
yeah, a lot of food for thought.
Yeah.
Let's thank Bill again. Awesome.